This is a Nobel Prize medal, a symbol of extraordinary scientific achievement. It belongs to Sir Alexander Fleming, who shared this honor with Dr. Ernst and Sir Florey for their groundbreaking work on penicillin. The story of penicillin's development was neither short nor simple, and it involves multiple researchers working tirelessly over several decades. In this video, we'll dive deeper into the journey of penicillin, from its accidental discovery to its transformation into a life-saving antibiotic that revolutionized medicine. Chapter 1. The Inception. The year was 1928. A Scottish scientist, Alexander Fleming, was working diligently in his laboratory at St. Mary's Hospital in London when he noticed something unusual. He had been growing a specific bacterial culture for an experiment when he went on vacation. Upon his return, he discovered that one of his Petri dishes had been contaminated with mold. We don't really know how it was contaminated, but it was a warm summer, so both the mold and the bacteria had a good condition for growth. Instead of throwing it away, he decided to take a closer look and noticed that there was a ring around the mold without any visible bacteria. It was like the mold had somehow killed off the bacteria around it. Fleming identified the mold as a strain of Penicillium and observed that it produced a substance he called penicillin, which had antibacterial properties. Back in the day, bacterial infections were a real pain in the butt. People would get infected, and there wasn't much doctors could do but cross their fingers and hope for the best. It was like playing Russian roulette with your health, and if you got unlucky, things could get really messy. Fleming had a lot of first-hand experience studying wound infection, having witnessed its brutal impacts on soldiers' wounds during World War I in France. At this point in the medical history, germ theory was pretty well established, and antiseptics were a viable treatment option on the battlefield. The prevalent use of antiseptics to prevent infection often backfired since they often killed the very white blood cells that fought infections, ironically inviting more trouble than relief. This first-hand experience made his discovery of penicillin all the more impactful and exciting. Theoretically, if this penicillin substance had been extracted and concentrated, it could have been used as a life-saving medication. However, at the time, he was unable to purify the substance and couldn't figure out how to make it in large quantities. So he did what most respectable scientists did. He wrote a paper about his discovery and let it sit on the shelf, gathering dust. To be fair, Fleming was really excited about his discovery, even though his peers were not really interested. Fleming knew he was on to something big. He even proclaimed in his penicillin paper that this substance would be a game changer in the world of antiseptic by inhibiting bacterial growth. This assertion was, in reality, a colossal understatement. The full magnitude of this discovery and its impact on medicine would only unfold in the years to come. To understand why, let's take a look at the pharmacology of this antibiotic. Chapter 2. The Science. Alexander Fleming's research focused on Staphylococcus aureus, a type of gram-positive bacteria. Bacteria generally have a cell wall, which can be classified into two main types, gram-positive and gram-negative. These bacteria are fascinating in their response to a specific dye. Gram-positive bacteria stain purple, while gram-negative bacteria turn pink. This difference is significant because it relates to the structure of their cell walls. Gram-positive bacteria have a thick outer layer made of a strong material called peptidoglycan. Think of peptidoglycan as a molecular fortress made up of two distinct sugar molecules shown here in green and orange, linked by peptide chains. This structure provides a protective outer layer similar to our skin or a turtle's hard shell. Disrupting this layer could be the key to defeating the bacteria. Penicillin doesn't directly break this wall, but cleverly prevents the bacteria from assembling and repairing this protective shell. Without it, the bacterial cell wall becomes vulnerable and collapses easily. Although the germ theory was fairly well established at that time, it was still not fully understood. In a sense, Fleming was fortunate because he happened to study a type of bacteria that was susceptible to penicillin. If he had chosen a gram-negative bacteria, he might have seen no effect, as these bacteria have an additional cell membrane that protects their inner wall from penicillin. This trait makes beta-lactams like penicillin more effective against gram-positive than gram-negative bacteria. With this deeper understanding of penicillin's pharmacology, let's rewind to the storyline. Chapter 3. The Oxford Team. This is Howard Florey, an Australian pharmacologist and pathologist. In 1935, Florey went back to his alma mater at Oxford to become the director of the Dunn School of Pathology, where he had a unique opportunity to build his own team. One of his first hires was Ernst, a Jewish chemist who grew up in Berlin, but then immigrated to England. At first, Florey and Chain wanted to do a big survey of the antibacterial properties of different substances made by microbes, so the team started their project with a literature review. And in 1937, Chain came across Fleming's original paper about penicillin. They thought that it was an interesting discovery and wanted to look more into penicillin and study its properties. So in 1938, Florey, Chain, and their team started trying to isolate penicillin from the penicillium mold, but they soon learned that it took a massive amount of mold just to get a little bit of penicillin. They were going through mold faster than they could get it. And this was when the team's biochemist, Norman Heatley, came in. He played around with a few different techniques for growing penicillium, but ultimately settled on growing the mold in a nutritious gel called agar, which served as a food source for microbes. His methods transformed the laboratory, which soon became filled with shallow and wide vessels of moldy agar. Heatley also standardized the measurement of penicillin's potency by observing the size of the inhibition zones around the mold. As the research progressed, the lab realized penicillin's remarkable efficacy in controlling bacterial growth and decided to try to turn it into a usable drug. And let me tell you, it was no easy task. First, they had to figure out how to grow the mold in large quantities, which was a whole production in itself. They had to find just the right conditions, the right temperature, the right amount of moisture, the right nutrients to get the mold to grow, and they had to do it all without contaminating the mold with other types of bacteria or fungi. Once they had enough mold, they had to extract the penicillin from it. This involved a lot of chemistry, purification, and time. After a lot of trial and error, they were able to produce enough pure penicillin to start testing it on mice. In their experiments, they tested penicillin on 3 groups of mice, each infected with a different type of bacteria. The results were groundbreaking. Nearly all untreated mice succumbed to the infections, whereas those treated with penicillin showed remarkable survival rates. These findings were published in 1940 in The Lancet, one of the most prestigious journals in medicine. To quote their paper, "The results are clear-cut and show that penicillin is active in vivo against at least 3 of the organisms inhibited in vitro." The implication was clear to them. This could be used clinically to fight infections. But as Florey pointed out, treating and curing infections in mice was one thing, but humans are roughly 3,000 times bigger and would need 3,000 times more penicillin. As a result, their lab was turned into a penicillin factory, production running 24 hours a day for months. After half a year of working nonstop, they finally had enough penicillin to start testing on real patients. Chapter 4. The Human Trial. By 1941, the team at Oxford felt they had enough supply to begin a trial on humans, and to the physicians performing the trial, it was like a miracle to them. The treatments for infection back then were pretty rough. If you got a scratch or a cut, they might put some antiseptics on it, but those were pretty harsh and could damage healthy tissue. In severe cases, they might have to resort to surgery, which was invasive and risky. And if you got an infection somewhere that couldn't be operated on, well, you were pretty much screwed. And then there were the drugs. Sulfonamides were one of the few options, but they had some pretty nasty side effects, including kidney damage, allergic reactions, and skin rashes. And they weren't even effective against all types of bacteria. More importantly, sometimes they didn't even work. The initial human test of penicillin began on January 17, 1941, with a woman suffering from terminal cancer. The goal wasn't to cure her cancer, but to test the tolerance and toxicity of the antibiotic. The first injection was 100 milligrams of penicillin, which was only about 5% pure, but because of the contaminants and impurity, she couldn't tolerate it very well. Even though she developed a fever and stiffness, fortunately, she tolerated the second injection much better. However, the first test of penicillin's effectiveness came about a month later, with a 48-year-old policeman named Albert Alexander. Alexander had developed a life-threatening bacterial infection after having a cut while doing yard work. Despite treatment with sulfa drugs, the wound worsened. The infection then spread to his eyes, and as a result, he became blind in one eye. So on February 12, 1941, he received an injection of 200 milligrams of 5% penicillin for 5 days. Initially, the treatment showed remarkable results. However, the amount of penicillin available was low, so the team had to recycle penicillin from his urine to extend the supply. Despite these efforts, the limited penicillin supply ran out before Alexander could be fully cured. As a result, he succumbed to the infection and soon passed away on March 15, 1941. They successfully prevented what seemed to be impossible at that time, but the scarcity of the drug was a challenging barrier. So, how would they address this issue? Florey later found the answer to that large-scale production question across the ocean in the United States. Chapter 5. Production in the US. At this point, World War II was raging in Europe, and the UK-based drug companies were mostly tied up with the war effort, so they couldn't help expand the penicillin production. Resources were scarce, and the constant threat of bombing raids made large-scale production a logistical nightmare. Realizing that they needed help, Florey and his colleague, Norman Heatley, embarked on a mission across the Atlantic to the United States. They later got in touch with the Northern Laboratory in Illinois. Together, they planned to increase penicillin production in 3 different ways. 1. Find a stronger strain of penicillium. 2. Develop a more efficient growing protocol. And 3. Improve the fermentation process to increase the purity of penicillin produced by the mold. The goal to find a more efficient growing method was soon realized when Norman Heatley discussed with the Northern Lab scientists his methods for growing the mold. They soon realized that using lactose instead of sucrose in the growth medium could significantly boost penicillin production. At the same time, they also completed the goal of refining the fermentation process. The traditional method of growing mold in shallow dishes was highly space inefficient. Inspired by the techniques used in the brewing industry, the Northern Lab designed massive, sterile fermentation tanks that could hold tens of thousands of gallons of nutrient-rich broth. Now the final breakthrough came in the summer of 1942. A lab assistant from the Northern Lab incidentally discovered a high-yielding strain of penicillin mold growing on a cantaloupe in a local market. This strain, astonishingly, produced 200 times more penicillin than Fleming's original mold, and after radiation treatment, it yielded a 1,000-fold increase in penicillin. Meanwhile, across the ocean, the remainder of the Oxford team were trying to figure out the chemical structure of penicillin to help the US team. By the fall of 1942, some UK-based companies had begun small-scale penicillin production. However, for widespread human treatment, they still didn't have enough materials, and Florey would ultimately require the industrial might of the United States' pharmaceutical companies. Unfortunately, Florey by himself couldn't convince US Big Pharma since there wasn't a ton of data showing that penicillin was safe and effective yet. Luckily, he had a longtime friend who not only helped him secure funding but also facilitated the mass production of penicillin. Chapter 6. Help from the US government. Now enter another player, the US Office of Scientific Research and Development, OSRD. The United States had recently started this department as a way to fund medical and scientific research related to national defense. When hitting a wall trying to mass-produce penicillin, Florey discussed with his longtime friend Alfred Newton Richards, who happened to be the chairman of the medical division of the OSRD. At that time, there was limited clinical data to support penicillin's efficacy. However, Richards saw a lot of potential in penicillin based on Florey's experiments, not to mention that he had a huge trust in his friend Florey. So Richards got representatives from 4 major US drug companies together, and in a meeting marked by a blend of persuasion and patriotism, he urged them to support penicillin research, hinting at possible government compensation. This collaboration marked a significant shift from the traditional model of medical research in the US, which had historically been conducted by government agencies. In October 1941, the first penicillin committee meeting brought together government officials and pharmaceutical company representatives. The initial discussions were casual, focusing on the production of penicillin for clinical trials. The tone shifted dramatically after the attack on Pearl Harbor, and the urgency of war heightened the importance of the project. Now, at this point in the scientific timeline, significant progress in the penicillium growing method was made at the Northern Lab. This breakthrough raised hope for large-scale production. At the same time, scientists at the US Department of Agriculture patented aspects of the production process, licensing them to drug companies without charge. Initially hesitant, the pharmaceutical companies now fully engaged in penicillin research, agreeing to share their findings with the committee. However, the OSRD maintained strict control over information dissemination to prevent public misconceptions about the availability of penicillin. Only 22 specially approved doctors were permitted to use penicillin, primarily for infections resistant to sulfonamide, such as those caused by Staphylococcus, Streptococcus, and Pneumococcus. Despite these restrictions, news of penicillin's remarkable effect soon reached the public in a dramatic fashion only after a couple of months. Chapter 7. Public Awareness and Mass Production. In March 1942, a woman named Ann Miller was admitted to a New Haven hospital. She recently had a miscarriage and later developed a severe streptococcal infection. Her condition then deteriorated despite conventional treatments with sulfa drugs and surgery. Luckily, one of Howard Florey's friends was also hospitalized there, so Anne's doctor asked him if he could request Florey for a sample of penicillin. Florey agreed and facilitated the delivery of 5.5 grams from a pharmaceutical company called Merck. Note that this was about half of the entire US supply, and apparently getting half of the stock to this woman in Connecticut was a really big deal. It required the government's approval in Washington and an escort by a state trooper. Ann Miller got her first dose of penicillin on Saturday afternoon, and by Monday, her temperature was back to normal. She became the first person to have their life saved by penicillin and lived another 57 years. Then, a few months later, another incident pushed penicillin into the limelight. On November 28, 1942, there was a big fire at a nightclub in Boston. Most of the survivors got rushed to Massachusetts General Hospital, where they were given sulfa drugs at first, but then Merck came through with literal tanks of penicillium mold. This would be enough to give patients a tiny dose of penicillin every few hours, but that was extremely crucial since burned patients often have damaged skin and, as a result, lack a protective layer to fight off infections. Most skin infections are caused by staph or strep bacteria, and the only widely available antibiotics at that time, sulfonamides, were not very effective against these type of infections. As the fire survivors received treatment with penicillin, its efficacy became more apparent, and penicillin was officially in the public consciousness. And as penicillin's reputation grew, the public began appealing to President Roosevelt for access. Despite the high demand, the limited supply meant that penicillin was primarily reserved for military use during World War II, given its ability to save soldiers from life-threatening infections. By June 1943, penicillin production was coordinated under the War Production Board, marking a shift towards mass production. From $20 per million units in 1943, the cost dropped to $6 in 1945, and further to just 10 cents by 1949. And just like that, the public could finally gain access to this amazing new medicine. The impact of penicillin on medicine extended far beyond its immediate therapeutic benefits, because its discovery ushered in a new era of antimicrobial therapy. The years following penicillin's revolution witnessed the discovery of a plethora of new antibiotics, each with its unique mechanism of action and spectrum of activity. Streptomycin, tetracycline, erythromycin, and a host of other antibiotics emerged as powerful weapons in the fight against infectious diseases, sparking an exciting wave of research and development in the field of immunology. And that is it, everyone. I hope that you learned something new today, and the next time you take an antibiotic, just remember that it all started with a Scottish dude forgetting to clean up his experiments and going on vacation.